So we're gonna go over viruses. First, the viral structure is made up a envelope protein, the envelope on the outside, which is the membrane. Viral genome, which is inside, which is made up of a DNA or RNA. Either could be single strand or double strand. And then the viral integument, which is inside the membrane of the virus. The envelope proteins could attach themselves to a host cell, but depending on the cell, it may or may not attach. Some of the proteins are very specific to either a living organism or to a actually like an actual host cell. And so different shapes um, are there in viruses. So there's the rod shape, which is the RNA, which is uh, covered by a capsid made of protein subunits, which would go in through endocytosis into a host cell. There's the icosahedral uh, shape of the virus, which is more like just a triangular prism type shape, and with the fibers, which would be the envelope proteins. There's a more complex structure, which is made up of the base with the base plate at the bottom, the tail fibers, which attach to the host cell, and the head capsid, which contains the DNA of the virus. And this would be more like in bacteriophage. And then the last one, which was as in the first one, is a membranous, has a membranous envelope with proteins that would attach to the host cell. And inside the envelope would be the RNA or DNA. So the reproduction of viruses, since they can't reproduce by themselves, they have to use a actual host cell to reproduce. In the lytic cycle, which is phage, the virus attaches to a host cell. It injects the host cell with its genome DNA, and the virus body is discarded and destroyed. Through the use of the actual cells, the host cell's reproductive system, the virus will reproduce and multiply within the cell, which then would later break out of the host cell. The host cell will be terminated, the virus will spread, and then it would attach again to another host cell. In the lysogenic cycle, the host cell isn't destroyed at first. So first, the virus would attach to a host cell. It would inject it with its DNA or RNA. The DNA of the virus would intermingle with the host cell's DNA, and it would go into like a hibernation phase, where it would stay silent for some time. And with the division of the cells through meiosis or mitosis, through mitosis, it'll just spread and it'll go unnoticed for some time. Then it could be activated by some sort of environmental switch, in which it would go into the lytic cycle, where it would reproduce. The virus would come back and reproduce inside the host cell. It'll break free from the host cell or host cells that have been made through mitosis, and it will go again and attach itself to another host cell to start the lytic phase again.